All right, everyone. Thank you so much for joining us today for the BC webinar series. Um, my name is Laura Newburn, and I am the coordinator for the Center for Bee Ecology, Evolution, and Conservation at York University, or BC for short, as many of you know. This webinar series has been organized by BC, and after the presentation, we will have a Q&A session. Please raise your hand as a participant. Which can be accessed along the bottom of your screen. And if the discussion, of course, extends past the hour mark of the set time, then you can no longer be present. Don't worry, both the presentation and the Q&A session are being recorded. And for speakers who give us permission to share the recording, they'll be posted on our YouTube channel in the next coming week or so. So as I mentioned, BC is the Center for Bee Ecology, Evolution, and Conservation at York University. If you'd like to learn more about us, please feel free to visit our website, yorku.ca/bees, or email me, the coordinator, bc@yorku.ca. Very briefly, we are an initiative that strives to advance research in the fields of bee ecology, evolution, and conservation. The mission of BC is to foster interdisciplinary, innovative, collaborative, and cutting-edge research. This research is then used for the advancement of knowledge and implementation of policy changes to help sustain pollinators globally. And overall, our goal is to apply our collaborative efforts to the development of policies and environmental management for the long-term sustainability of bees and, of course, the vital ecosystem service they provide. If you would like, if you would like to keep up to date with all that goes on at BC, please join our global listserv. Send an email to listserv@yorku.ca with the following command in the body of the email: subscribe bees, and then your first name and your last name. If you are a York University researcher or collaborator who has not investigated BC for membership, please visit our website under the membership tab to learn more about the benefits of becoming an associate or to apply. You can see below here we have a small selection of our happy BC associates, some of whom are even out in the field collecting specimens or doing research. So we have an exciting lineup of talks for our fall semester BC speaker series. This is a series that takes place once a month on either the second or third Friday of the month at 11 a.m. Eastern via Zoom. Typically, it is through the Zoom webinar platform, but for today, we're changing it up with a Zoom meeting. We have one more talk in our fall semester, which will take place on December 13th with Dr. Christopher Starr, and we will have more information on our speakers for our winter semester coming soon. So please keep up to date on our website, or as I said, subscribe to our listserv because we will publish that information in our monthly newsletter. So the logistics for today. As I mentioned, we are using a Zoom meeting platform, so please, we ask that you have your camera and your microphone turned off during the presentation. And at the end of the presentation, if you'd like to ask a question, please raise your hand and I'll call on you. And then you can turn on your camera and your microphone if you'd like to ask your question. Also, feel free to use the chat box during the webinar, but we do ask you to use it sparingly for general comments as it can be distracting for our speakers to have a bunch of notifications popping up. If you have any trouble, please send a private message to me in the chat, Laura Newburn, or email bc@yorku.ca. As per Matt's preference, this presentation will be recorded and posted on our YouTube channel almost immediately after the presentation within a week or so. Now, speaking of our YouTube channel, we have several previous talks posted there, including previous webinar series like our BBST series, the Associate Speaker series, and several talks from our past BCons. And in fact, the first round of videos have been posted from BCon 2024, which took place earlier in October. They were posted in about a week ago, actually. So we invite you to check them out. More videos will, of course, be posted from our BCon 2024 presentations. So we invite you to subscribe to our channel so you'll be notified when the next videos are posted. Before we begin for today, we would like to start the day with a land acknowledgement. We recognize that many Indigenous nations have longstanding relationships with the territories upon which York University campuses are located, that precede the establishment of York University. York University acknowledges its presence on the traditional territories of many Indigenous nations. The area known as Takaranto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis. It is now home to many Indigenous peoples. We recognize the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. If you would like to know more about the history of land that you come from, or where you're joining us from today, or where, let's say, you collect your research samples, this website, native-land.ca, is a fantastic resource for exploration. It was initially a Canadian initiative, but it now has worldwide coverage. And it is now my pleasure to introduce our speaker for today, Dr. Matthew Betti. He is an associate professor of math and computer science at Mount Allison University, and his research focuses on the mathematics of disease transmission and mathematical modeling of ecological processes. Thank you very much for being with us today, Matt, and I invite you to share your screen.
Thank you for the invitation, and thank you for coming, all of you. This talk, I watched a couple of the BC talks on YouTube to prepare, and this might be a little bit different because, again, like was just mentioned, my work is mostly in mathematics, and I try to apply some of that mathematics to pollinator systems. I also do some disease transfer in humans and bees, and it's weirdly not too different from one another, and more broadly to ecological and evolutionary problems, specifically those currently that affect the Maritimes, since that's where I'm currently living. So we're doing a lot of work with turtles, particularly right now. But today I'll talk to you about honeybees. So this is going to be a little bit different. It's not necessarily about a single project that I've done, and more just an overview of what mathematical modeling is and how mathematical modeling can be used to provide insights into the complicated life patterns of complicated ecologies of pollinators, and specifically of honeybee populations, and what those insights might be able to tell us in terms of conservation. So one of the problems that historically, I mean, it affects a wide range of biology, is that you want to study things that are wild. You want to study the world, the natural world as it is, and yet we often have to do this through highly controlled environments. So on the left of your screen, that's what a natural honeybee colony will look like, or a wild honeybee colony. And mostly what we study are these man-made hives. And in fact, in North America, I believe, I read somewhere once upon a time that there are not that many wild honeybee colonies. It's very difficult for them to survive, especially up here in Canada. And so this can cause a discrepancy on sometimes what we see, what we observe through experimentation, and what can be applied to wild populations. There's also differences between, and probably preaching to the choir here about, the issues affecting honeybees as livestock versus wild honeybee populations. And it can be difficult through experimentation to differentiate the two of these things. So traditionally, what we do is in the sciences, we design experiments, we make observations, we analyze data, and we draw conclusions from that data. It's worked. It works very, very well, but it can be expensive. Sorry, I don't know if you can see the little zoom thing on the side of my screen, so I'm just going to hide it just in case. Sometimes this can be very expensive, and oftentimes what we see are correlations. And then when we're in that making conclusions part of a project is when we have to hypothesize causation, possible causations for the correlations that we see, and then we have to go back and design new experiments, make new observations. Um, and oftentimes this is exacerbated by the fact that we're limited by the data that we have. And without rerunning new experiments, tweaking experiments, um, it's often difficult, especially to find these causative factors. And this is somewhere where mathematics can actually help quite a bit. Oops, why won't I move? So when we introduce these kind of theoretical frameworks through mathematical modeling to ecological problems in general, specifically, I mean, keep in the back of your head that we're applying these, at least today, to pollinators and specifically to honeybees, and even more specifically, honeybees as livestock. Um, Oftentimes, these mathematical models can give us insights that can inform our experimental designs and prevent there's no such thing as too much data, but there might be too much data for what we're trying to do. And so, we can limit costs and time through informing experimental designs. We can circumvent some of those costlier aspects of experimentation by, trying to develop a theoretical framework that can at least give us a good idea of what is important to measure and what is not important to measure. Um, it can also save us, I mean, for the most part, I know it's less and less true with every passing day, but most mathematics is done now uh, through a computer, or at least assisted by a computer, and compared to making big experiments, running a computer is relatively cheap. So we, save on costs that way. And what I think is sometimes the most important thing is that we can provide through first principles, and just through logical and rational arguments, we can sometimes provide causative relationships, or at least insights into possible causative relationships um, for the phenomena that we observe in the real world. So that's my little cell on mathematical modeling, my high level cell. And there are many types of models, and as scientists from any walk, we use them all the time. So most scientists would be very familiar with statistical models, where we use observations to determine relationships between variables. Oftentimes, given the data, we will use uh, linear relationships. If we can, if we have enough data, sometimes we can extend that a little further into the nonlinear spaces. Oftentimes, what we can see through data is correlation, right? And then we have to build theory to try to scratch away at the causative relationships between variables, and then, like I said, do more experiments, more carefully designed experiments to get to those causative relationships. The other big type of modeling is mechanistic modeling, and that's kind of where I live. And I saw Jane Heffernan's picture on those introduction slides. That's where she lives too. These mechanistic models are usually more mathematical in nature. They're usually a little bit more deterministic. They can have some randomness and stochasticity in them, but they're usually derived from first principles. So using observations that we have and phenomena that we know, we build these interactions kind of in a theoretical uh, microcosm. And using this, I think one of the strengths of mathematical modeling, and hopefully I'll convince you that these are some of the strengths, is that we can develop and determine the causative relationships between the outcomes that we observe empirically and what we know is generally what is happening with different populations. And so I kind of live on that left-hand side in the mechanistic world. So in terms of modeling, there is a golden rule no matter what you're doing with modeling. And when we get into how we tune models and build models for pollinators and honeybees, this will, I ask that you keep this in the back of your mind, because we do see a lot of, as a mathematical modeler, it's a very interdisciplinary thing, mathematical biology. We interface with probably the most theoretical of theoretical people in mathematics, where there is no real world that they concern themselves with. And then on the other side, in applied math generally and in mathematical biology, we interface with biologists who live in the real world, for lack of a better term. And straddling that line can be a little difficult, but there is one rule that we like to follow, and that's simplify things as much as possible. And things should be as simple as they can be without being too simple. And it sounds like something that shouldn't be true. It shouldn't be able to be done. But this is where applied math kind of distinguishes itself, is in determining how much we can simplify, what assumptions we can get away with without compromising what we're trying to study. Um, And again, I ask that you keep this in the back of your mind, because I will say things about bees in that are generally true, but they will also, alarm bells will start to go off and say, well, that's not always the case. And it's not, but I live in a world where we're trying to be as general as possible and as simple as possible to get at some key truths underneath. And so this is a diagram that Jane Heffernan originally had made, and I've adopted for this talk. And mathematical modeling is all about finding this balance, right? There is what we're looking for is biological insight into some problem in ecology, in evolution, in conservation, something. And the tool we have is, at least in my realm, is in mathematics. And mathematics, because it lives outside of the real world in its own little space, can get very complicated. It's not bound by the rules of physics. It's not bound by the rules of the universe. It is, in fact, bound by nothing except your imagination and a couple axioms that make sure that we're not doing anything contradictory or illogical generally. But the goal is biological insight. And if we take math that is too simple, we might not get enough biological insight to be valuable to the problem at hand. And if we get too much, there is a world where you can make things so complicated, you can basically rebuild the world as we see it in a computer so that you can simulate it and control everything. And then you're no better off than you were working in the real world, right? You don't gain any new knowledge by being able to reproduce the real world with all of its nuances in a computer, because we already have a very difficult time studying the real world. And so the goal is to kind of be here, where we have mathematics that is complex enough to capture all the features of the system that we're trying to work with, but we also want to keep that um, system mathematically tractable so that we can use a lot of the tools we have in mathematics to gain some of those biological insights. And I think it's an open secret among mathematicians and less open among scientists at large. We're not good at anything that is nonlinear generally with math. I would say the big portion of mathematical theorems and of mathematical knowledge is how to turn things from nonlinear to linear systems. And once things are linear, we know how to work with them. So most things in the world are nonlinear. Most things in the universe are nonlinear. Most things in mathematics, pretend universe that they live in, most of those things are nonlinear. And so we have a very narrow space that we can work in, where we are trying to keep things mathematically tractable, mostly by keeping things as close to linear as possible. But we also need to capture some of that nuance of the world around us. And the way we do this is through assumptions, right? We assume things about our system that shouldn't, at least using rational arguments, shouldn't affect um, what we are trying to study and generally are independent of what we're trying to study. Generally, you end up on this side of the graph, on this right-hand side here, of too much mathematical complexity and not enough biological insight when you have too many assumptions or when you don't have enough assumptions. So all that is just a precursor to justify my way oversimplification of a honeybee colony so that we can gain some small insights um, into some of the phenomena that we may observe. So when we simplify, I'm going to simplify the honeybee colony by saying that there is a queen. That queen is responsible for all reproduction in the hive. Now, here's where my first assumption comes in. Well, I mean, that was already an assumption, right? If we're really being technical about honeybees, because there are uh, worker bees that can lay eggs, mostly drone eggs. So she's not responsible for all, it's like all with an asterisk, all the meaningful reproduction within a hive. We're going to assume that she spends all day, every day, just laying eggs, fertilized or unfertilized. Um, Again, we're not bound by time in mathematics, so there may be periods of time when the queen is not laying eggs. Our version of time is just going to pause at those moments. And otherwise we're only counting time when the queen is laying eggs, basically. Um, and we're going to assume that without her, a colony descends into chaos, mostly will eat itself and die. Uh, this is so that we don't actually have to deal with the queen in a lot of situations. We're going to take the queen as a basis. We need the queen for a healthy colony to survive. And that way we can just kind of treat the queen as sort of the gas pedal to the car that is our colony, the main driver of the continued existence of a colony. We're going to simplify the worker bees as the backbone. We're going to assume that they're all sterile so that they don't lay eggs. They don't really contribute in a reproductive way to the colony. We will, and this is one of those things that we don't want to assume and that we want to model because it's pretty important to uh, a colony that the workers maintain the eggs and larvae and rear them to adulthood. They also build the hive itself. The models I'm going to show you today don't really focus on hive integrity. And that's mostly because these models were built for um, bees as livestock, where yes, in the wild, the workers would be the ones building the hive, creating the cells, patching hives. The integrity of the hive becomes more important when you're dealing with wild bees, whereas in a livestock or in a managed context, you have humans to help the bees with that. And so it becomes less important. And then the workers will eventually, we split them into two kind of subcastes of bees, where in their early days, they will mainly be responsible for egg and larva rearing. And then as they get older, they will start to go out and forage. And you'll notice a lot of behaviors of the workers here that we just have not accounted for. And because if we're trying to get some very general idea of how a colony functions and some of the consequences of changes to, say, um, what age the workers start to forage at, or the effectiveness of their brood rearing. A lot of those other smaller tasks that the workers have been observed to do are less important. And so this is, kind of trying to draw that line and allow for enough complexity so that we can draw some conclusions without conflating other behaviors. The drones, when we model drones, we model them very, very simply. They eat, they mate, and they die. we don't really, you know, use them for much more when we're modeling. Uh, if we wanted to build models about temperature regulation, then yes, we would um, talk more about drones or even wild colonies where their you know their impact on the food source may become uh, quite important in the survival of a colony. But if we're thinking about managed colonies, we can always, you know, for better or for worse, supplement the food sources of managed colonies to the point where the drones aren't necessarily going to um, eat the colony to extinction. And you know, there are mechanisms, again, glossing over, really oversimplifying a colony. And I hope to show you that this simplification is not necessarily without merit. But yes, so when we talk about drones, we're going to talk about them very simply. So one of the type of modeling that I like to do and that I find is very useful um, are called compartmental models. So like we just did, generally to build compartmental models, we break up a population into subgroups or subpopulations, and then we try to describe in words, in mathematics, uh, how those groups or those subpopulations behave and how entities transition from one to the other if they can, or how one group affects another group. Um, so we, you know, very basic questions that we ask are how do you enter a group? How do you leave a group? What effect does your group have on, what effect does group A have on group B and vice versa? Uh, so like I just described, we're going to just assume that the queen is ever present because the colony requires a queen to survive. So we immediately remove the queen as a compartment. We don't need her necessarily. She's there, ever present. And we break up our population or our colony into three subpopulations. One we call the hive bees, who are, again, responsible for brood rearing. We have the foragers. These are the older workers that go out and gather resources for the colony and bring them back. And then we have the drones that are just kind of doing their own thing, um, eating, sometimes mating, and dying. And generally, we don't often, unless we're dealing with multi-generational cycles, we kind of move that mating thing into the background because, again, we're assuming a queen is ever present and ever able to produce eggs that will become viable workers and viable drones. And so the reproduction, unless we're looking at very long-term studies or simulating and modeling very long-term phenomena, the the drones are really just responsible for taking up space and eating. And so now what we want to ask is how do you enter groups and how do you leave groups? So generally, if the queen will lay eggs and then those will be reared by the what we call the hive bees to maturity. And if they are female, they will become themselves hive bees. So any surviving female brood, once they reach maturity, will become a hive bee. They will start to uh, manage the brood. And as they age, we will transition our hive bees into foragers. They mature into foragers. We don't need this arrow. There's no mechanism from which you become a forager and then become a drone, right? It's an impossible mechanism, so we ignore it. Um, Foragers die of old age or environmental hazard when they're out in the world. Um, the drones will die of old age, or if they mate, or if winter comes along. So when we look at, say, seasonal models, right, the overall dynamics of a hive is changed with the seasons, and it's something that we have to consider if we're doing any modeling of phenomena that last longer than one, say, active season of a honeybee colony because the dynamics change so much. Um, so, over winter, basically, as winter approaches, the worker bees will forcibly remove the male bees, the drones from the hive, and they will be left to die outside. Um, how we create drones is through, well, same way we create female hive bees, we create drones through the surviving male brood. So what we do is assume that the queen is laying a certain number of eggs per day, and some percentage of those are female and some percentage are male. And that percentage can change with time if we like, or we can take an average over several months and see if that uh, is good enough to get some insights into colony dynamics. What you'll also notice here, so I'm just keeping an eye on the time, is that we don't have a way for hive bees to die. And this is another one of our assumptions when we're in our framework, when we're dealing with these models. We generally assume that bees within the hive, especially a managed hive, are quite safe. And so the death rate of a bee who never leaves the hive is going to be much, much smaller than the death rate of a bee who is out in the world and exposed to the elements and is objectively older than the hive bees. And so, this assumption is something that you could relax if you wanted to, if it was important to the study at hand. But generally, it's such a small proportion of bees, um, unless they're sick, that we avoid it. So what do we do with this now? I mean, we have blocks, we have arrows. One of the natural mathematical tools to use compartmental models are called differential equations. And we use them to describe the way a population changes over time. And they help us predict average behaviors of a population over time. And then it's, those predictions and the control of these arrows and these boxes through differential equations that allow us to gain insights, ecological insights into pollinators. If you don't know what a differential equation is, I will show you some, but very generally at a very high level, we use this all the time in the world. It's, if there is some sort of event, there are two ways to track whether you have too many people in a room or not. Event spaces have a capacity. You can either send somebody around consistently to count all the people in the room so that at different time steps, you know exactly how many people are in the room. That takes a lot. Or you can do it once. You can count once, preferably when there's nobody there, so the count is easy at 0. And then you can station somebody at the door who can count people that come in and subtract people that leave. And that is effectively what a differential equation does. It's not counting, it's not um, um, qualifying or quantifying behaviors directly. It's quantifying everything as a flux. So it's the equivalent of stationing somebody at a door, and it makes things much simpler to do than to try to count things and observe behaviors directly. And so I think I've got about 10 minutes left. So we'll run quickly through what differential equations are. So you'll see on the left-hand side of these equations, these are our populations, H, F, and D. And the d by dt, these are a derivative, and it's just this is how they change through time. And you can read this as here is a description. The right-hand side is a description of how the left-hand side population changes through time. And so you can see here that we have some survival function, right? The survival of the brood will depend on the number of bees that are available to look after the brood. It will depend on the foragers through food. Foragers are the ones who go out and collect resources. So the more foragers you have, the more resources you have. And another simplification here is this idea that you have a a positive relationship between the number of foragers you have and the number of resources available to your brood. Resources mostly being food, right? And then their survival also depends on the drones because the drones are taking away food for the most part. We have this R function, so we lose hive bees when they are recruited to foraging. When they get old enough, they leave after a certain amount of time. They leave their duties as a hive bee and they become a forager. And then they forage until they die, right? We lose foragers. The negative sign means we lose. We lose foragers at some average death rate. And so I won't go into the mathematics here, but very generally, this rate mu, 1 over mu would be the average lifetime of a forager. So if a bee on average lives for 40 days and they spend the last 2 weeks of their life foraging, um, mu would be 1 on 14. And the drones, you see this 1 minus p and the p here. Well, this is the proportion of eggs that are female and male. Um, and there is this survival function here too, because the male brood still has to survive. They still require resources. And the drones die at some average rate, which is usually quite a bit smaller than this rate. But because we don't have anything to compare it to, right here, we're comparing this number and what a number here that doesn't exist up in the H equation. There is a number there that doesn't exist. Um, we have two things to compare to, and one is much smaller than the other. So one is we can effectively treat it like zero compared to the other one. Whereas here we have nothing to compare it to, and so we put the number indirectly. And so what can this tell us? Oh, sorry, I forgot I had all this in here. For the sake of time, I thought I deleted all this color, to make things more interesting. I didn't. So another simplification we can make is, okay, well, maybe those drones aren't eating so much food that, you know, it's affecting the survivability of the brood. And so we say that, the survival of the brood depends on the proportion of your hive that are rearing the brood and also the proportion that are bringing in food. And they're multiplied together. Both of these numbers will be less than 1, right? So the survival function is a survival probability. We can also specify using a function what recruitment looks like. So very generally, because of the uh, pheromones released by foragers, uh, they can either recruit more bees to foraging, or if there are too many foragers, the pheromone that they produce will discourage other bees from maturing and leaving the hive. And so this 1 minus sigma this sigma is 1 over the proportion of bees that we expect to be foragers at any given time. Anything more than this, and those chemical reactions will start to discourage more bees from becoming foragers. Right? So generally, recruitment decreases as the forager population increases, and we assume survival requires care from nurses and food from foragers. The drones work in the exact same way, except we add this little K down here because they require a bit more food. Uh, so what do? This is about the simplest model we can have to describe a honeybee colony. And the first thing we do is try to match this to observed data. Right. So when we observe a colony, usually we are observing them at what we call an equilibrium state. So that colony has an average number of bees in it, and that average number will stay relatively constant, at least while the dynamics are the same. So in early spring, we should expect a certain colony size. The same thing when it comes to summer, fall, and then winter, we should expect a certain colony size, although the dynamics drastically change in winter. So we would need a different model. Right, so that generally what we look for is these unchanging average populations, and they can give us, that's what we can generally observe in the real world, and then we can tune some of these parameters so that we are matching what we see in the real world. Right, and so when we tune parameters, if we take certain parameters, like here is a non-biological example of an output of our model. We have 35,000 drones here, and the population is less than 10,000. And for a managed colony, that is unrealistic. We'd almost expect this to be flipped, right? We would expect, maybe 30 to 40,000 female worker bees and somewhere around 10,000 drones. So we would tune our parameters until we get there. And so what we can see from this model is that generally, in order for our population to be realistic, our foraging population it has an upper limit. If the foraging population is bigger than this, then we are going to get something unrealistic or unsustainable in a realistic environment, like a colony with 40,000 drones in it, right? And so this is one of those insights we can get mathematically, where our foraging population has an upper limit. And then we can analyze what this upper limit is to try to gain some insights that are relevant biologically. So foragers, the longer lived foragers are, the fewer foragers a colony can sustain. If they're longer lived, that means that from a biological or chemical perspective, they're going to produce more of that pheromone that discourages recruitment, right? They're going to produce that for a longer period of time because they're living longer. And so this makes sense biologically. If they're living longer and they're bringing in food, and we have this mechanism that we know is true, we don't need as many of them. But we also see that if we have a lot of drones in the population from this model, right, the more drones we have, the fewer foragers we can sustain in a population. And that's what that P tells us here. This L here is the laying rate of the queen. And so if the queen is laying more eggs, yeah, we can sustain a much higher forager population because we have this more consistent replacement of worker bees. Right? And so using this, we can actually estimate a healthy ratio of nurses to foragers. So by solving these equations up in the top left-hand corner, we can get what we would expect a healthy number of hive bees to be in a colony. And you can see that this depends here on the number of foragers. And so if we could rearrange this to determine the healthy ratio between nurses and foragers in a colony. And again, this is something that can help beekeepers at the ground level if they have a rough estimate of how many bees are leaving the colony on any given day and a rough estimate of how many bees are in the colony, we can see if that is healthy. And if it's not healthy, we may end up in a situation like this, right? But it gives us something that we can look to before this happens. So we might be able to mitigate. So that's a very simple model. There's some mathematical complexity there, right? But we can probably do better. We get a little bit of biological insight there. We have a roughly an upper limit on the number of foragers we can have in a healthy colony, and we can determine a healthy ratio, a healthy average ratio for a colony. So there are some ways we can expand this model. One of the ways is to say what happens if we have disease in a colony. And again, this works exactly the same, except now we're splitting our three groups further into those that are healthy and susceptible to disease and those that are infected with a disease. So this disease, it's a general disease. It can be anything from varroa mites to nosema. The only diseases this doesn't really capture this type of model is like a American foul brood or any other disease of the brood, right? And this just, again, you can see very clearly the mathematical complexity increase here. We now have 6 equations. There are these interactions between infected bees and healthy bees. That's a proxy for, say, a mite, moving from one bee into the brood and then onto what was a healthy bee. Or with nosema, just the spores created by the infected bees and left around the colony being absorbed by healthy bees and infecting them. Right. And then we see here that yes, the infected bees can die now, right? There should be a minus sign. Sorry. The hive bees, the death of them becomes significant now, and infected foragers, infected drones can die. Um, it reduces the lifespan. Disease reduces the lifespan of them. And we can see that this can lead to catastrophic failure of a colony, right? This arrow here is when we introduce disease into a colony, and it can spread fast. And if it's particularly lethal, um, we can see a catastrophic decline within, say, a month and a bit of, uh, yeah, of a colony being infected, right? And this is for that extra mathematical complexity. This is an extra insight that we gain. We can also see that, as we start to tune, there are different things that can happen. A colony can collapse completely, a colony can become unstable, and that instability, if we have a change in behaviors of the colony, like say fall to winter, um, that instability can lead to death, an overwintering death of a colony. And we can also see that, there are, these things are affected differently. The timing of when a colony gets a disease affects how well it can survive. So this delta T on the bottom is the days before the onset of winter um, that a colony gets infected with a disease. And we can see that they're particularly susceptible if a disease starts to run rampant in those final days. And that's what generally causes, at least mathematically, this kind of instability in a colony that leads to death. Right. So we add some mathematical complexity, we gain some biological insights. But it's not the only way to expand our model. There are other ways, and I'll promise I'll finish up in 2 minutes. Uh, right? We can also study things like the problem of diploid males in managed colonies. Diploid males come when there's too much inbreeding among queens and drones, and then queens start to produce these female bees that have matching sex alleles, and so they're meant to be uh, worker bees, and they come out as diploid males, which can be kind of catastrophic to the colony. So we can do this. We can look at this um, by adding a diversity term here. This U is a measure of diversity of the number of sex alleles in a sort of a meta population of bees, right? And if we add in terms like this, we see these complicated mathematical formulas pop out. And I don't expect this to make sense, and don't worry if it doesn't. It's just a bunch of Greek, quite literally. But what it shows is that, if we have too much inbreeding, which can be caused through selective breeding unintentionally, we can see that we end up in this situation where we might have too many drones. And mathematically, this colony can be sustained with this many drones, but practically, and this is where we bring it back to the real world, practically, it's unlikely for a colony like this to survive. And so the insight we gain here is that there is we get this minimum value of diversity that is required, right? U equal 1 means that it's completely diverse. You will never ever get a matching sex allele. U being 0 means there is only one sex allele in the population, and everybody would be a diploid male. And so there's only so much inbreeding a meta population even can sustain. And so again, we add some mathematical complexity, we gain some biological insight. And these insights can inform, how that we should be careful. We should be careful when we are breeding queens, that we don't use populations that are too close together. And oftentimes with managed colonies, this is what happens, right? The drones are coming from a nearby colony to the queen, and we will sometimes end up with large amounts of diploid males, right? And again, this is showing same thing in a slightly different way, that this U star is also affected by, if you remember what mu was, it was the death rate of foragers. So if the bees are in a hostile environment, right, where their death rate is increasing. This graph is kind of hard to read, but basically the lower this number is, the longer they are living. And the longer they are living, the this U star is the inverse of the U on here. And that just comes from this Greek that I told you not to pay too much attention to. Uh, so basically, if they are dying faster, there is less, uh, the colony or the population at large is less able to uh, deal with the effects of inbreeding. And so this is, again, one of those insights we can see by these interactions that we may not, it may be very difficult to observe in the real world. We can mathematically point to maybe this is something we should look into. So I guess this was mostly a sell on why mathematical models are useful. But I hope I did convince you at least a little bit that there are insights we can gain here. And I hope I gave a little bit of, you know, insight, a few insights that we can gain through mathematical models for pollinators. One of my favorites being this interplay between the hostility of an environment and a colony's ability to manage the effects of inbreeding. But yeah, I think I'll leave it there. Sorry, it was a little longer than I thought. And I guess we can take questions.
Awesome. Thank you so much, Matt, for your talk. So I'll turn it to the participants now, our audience members. If you have a question, please feel free to raise your hand and I will call on you. But I guess just while people are doing that, I had a small question.
Yes.
So when you were talking about disease modeling, you
Yeah.
mentioned that in your particular set of equations, it wasn't good for estimating foul brood because, of course, you weren't taking that into account in the equations.
Yeah.
Does that is it more complicated to add in the term of how a brood infection would then impact the worker bees, the nurse bees, etc.? Or is it just
Yeah.
because it wasn't in this particular set of equations?
It's just so it's not in here, but like we can do this. We can imagine a world where we do this by adding that group to our population, right? We can have, if you want brood, susceptible and infected brood, you could have a dB by dt, right? Which would then allow you to build an equation for the brood. And then you can talk about how that might spread through the worker bees who can move around and how that foul brood might spread to new eggs or new patches of brood through the worker bee. So you can add equations, then you would have 8 equations with a lot of terms in it, but you might be able to remove some other ones. If we're talking about foul brood specifically of the worker bees, because I guess they are, I think, objectively the more important of these classes of bees. You might not need to model the drones, right? You might be able to bring this back down to 6 equations, kind of lower that sort of mathematical uh, complexity and still gain some insights, like, how quickly should you deal with the American foul brood before a colony is beyond the point of saving, right? And these are things that beekeepers, I think, know intuitively. When that's true, I think researchers can do experiments to determine that. There's just another avenue to kind of get at the same answers, and this one we can do kind of on the cheap. Uh,
Awesome. And I guess a follow-up to that, with these mathematical sort of models, do you take them out into the real world and then say, well, let's count the time of when foul brood is going to be a problem? Does it match with the mathematical model or is it very
yeah.
different?
So yeah, we do. Like again, I was focusing on the math here, but yeah, generally we will at least scour the literature, right, and make sure our models make sense with the literature, and we will talk to biologists and make sure that what we're seeing makes sense with what they observe. Because sometimes when you observe things, it's qualitatively easy to say, yeah, that makes sense. But there's so much noise in the real world. It's hard to get to the underlying mechanisms why certain things happen. One of the things that I didn't show here, but what we one of the insights that is gained in a lot of these types of models is this 1 over this recruitment function here gives, much like the 1 over this death rate gives the average lifespan of a forager, 1 over this recruitment function gives the average age when a bee starts to forage. And you can see that instability in that average age to foraging will lead you, in many cases, not just in disease cases, to situations like this that will lead to the long-term destruction of a colony. And it's a very early indicator because sometimes you will see it back here before there's any kind of instability or collapse.
Oh, okay,
And so
very
that's one of
cool.
those insights that we can find. And then, we give that to people. They go out in the field and measure and like, is this actually something that is this a useful measurement that we should be putting more effort into? Um, and I think it is. I think there are papers out there where they have looked at that.
Okay, awesome. So I see that we have one question from our audience. Sarah, feel free to unmute yourself and start your camera if you'd like.
Well, thanks. Sorry for the loudness. I'm just on the bus. Can you hear me okay?
Yes.
Yep.
Okay, cool. Thanks for the presentation, Matt. It was great. I was just following up on, sorry, Laura's question about when you go to validate the models. Can you give us
Yes.
an example of how you would adapt the model after the validation? Like
Yeah.
if
So a
or
lot of
whatever?
yeah. So a lot of it is these parameters, these numbers that we have here, like this P. Um, where are we now? Where are we? Sorry, this W, this B, this alpha. These are things that either people have done experiments and have found rough estimates. So this sigma here, if we look in the literature, this is something that's fairly well established, right? This is we would expect about a third of our worker bees to be out foraging at any given time, or be having the ability to forage at any given time. And so we would put that in and then try to tune the rest of these parameters to match key metrics that we can find either in the field or the literature. One would be, we can determine on average how many brood are surviving to adulthood. I mean, it's not necessarily always easy to count brood cells, but we can get an estimate. And so we would kind of tune this W and this B so that those things match. And this, again, for the results I presented here, where they were much more qualitative, that specifically matching it to numbers in the real world is slightly less important, right? It doesn't matter what the numbers are, this relationship is going to hold. So as long as the mechanics of the model are describing the real world, then if you can measure these things, you will see what your upper limit here is on, say, a foraging population when we're accounting for uh, uh, the queen putting out a certain proportion of drone eggs and a certain proportion of uh, worker eggs. Now, what we would do is go out and try to measure what P is, or find studies where they have measured roughly what P is, find mu, find L, and then see if our upper limit of foragers that our model gives matches, say, the number of foragers measured by individuals in the real world. And if it doesn't, we go back to the drawing board, right? It's very cheap. This gets written on a whiteboard, these equations, and it's very cheap to turn around and say, okay, this survival function doesn't work. This, we thought it would work, but it doesn't work. What are we missing? What piece, what contributes to the survival of brood that we don't have in here? And you add mathematical complexity slowly, slowly, until your results start to match at least what's seen in the literature. Now, a more complicated way to do this is to take data from the literature and then try to fit your model to that data. And that gets very noisy and very complicated very fast. So if we're looking for these sorts of overarching qualitative insights, then we don't necessarily have to do that. If we want to be specific and predictive, yeah, we would do something like that, or, replace some of these parameters with entire distributions, and then do a literature review or a meta-analysis of what that distribution of that parameter should look like. Does that kind of answer your question?
Yeah, cool. Thank you.
All right, wonderful. So I don't see any more hands up in the audience. And we're a little bit over time now. So I'm going to wrap this up. I'd like to thank you so much, Matt, for joining us today and for talking to us about mathematical modeling. I'm sure that everyone here has learned a little bit about it. I'm sure that if anyone thinks of a question later on, you would be more than happy if they emailed you and for a follow-up.
Absolutely. Absolutely. Always
Thank
happy to
you.
talk.
Awesome. And with that, I'd like to remind everyone that next month we have our final speaker series talk in the fall semester. It'll be with Dr. Christopher Starr, who's our visiting scholar from the University of the West Indies, Trinidad and Tobago. So please join us that day. You can sign up on our website, and I will send you reminder emails about it. So, thank you very much, everyone, for joining us today, and we look forward to seeing you all again next month.